Shiley. And hi, everyone. It's my first time being here in the in-person Lunch and Learn series, so it's really exciting. I see how it goes. And thank you for coming, and thank you all for showing up online. So I guess I'll introduce myself just a little bit. I am currently assistant professor at Neurosciences Department at UCSD. I've been with ADRC since the very beginning, since I joined, so being a long-term citizen. And I'll talk a little bit about my career trajectory here. So hopefully that will resonate with some of you. But my lab, this is my lab website out there. And my lab is at Leach Tech Building. And I'm a completely wet lab-based basic biologist. So I don't do clinical work. I use ADRC resources a lot. I use ADRC samples a lot. But basically my bread and butter is animal models, cell cultures, and some of the cellular molecular tools. Oopsie. Let's see. There we go. So a little bit about me. I was born and raised in China, and I did my undergrad at Peking University in Beijing, China. And after that, I went to University of Wisconsin-Madison to pursue my PhD in genetics, and I studied fly genetics. And after that, I went to UCSF in the Bay Area, Gladstone Institute, and I did my postdoc with Dr. Li Gan, and I studied Alzheimer's disease, and that's where my career started. I was awarded a K99 award back then, and that was the beginning point of my faculty career. And then I relocated down to San Diego to start my faculty position here right before the pandemic, as you can see. And let's see, yeah, so I guess in terms of my career goals, I wanted to study the mechanism of the pathophysiology of Alzheimer's with a special focus on tau. And I want to come from a more sort of unique perspective, which I'll talk about in a bit. And moreover, besides doing the science, I also try to build a productive and inclusive and collaborative research program so that I will help other people with their career. So a little bit about my trajectory here. I mentioned I had a K99 award, but it's a kind of unusual transition, I would say. It was broken, as I call it, because I basically moved down to San Diego before securing a job. So I was kind of at the last phase of my K99. And because of the hiring took longer than expected, I ended up not getting the R00, which is pretty unheard of. However, thanks to the ADRC, I was able to turn lemons into lemonade. In this case, I got a P30 a re-entry supplement award, which really helped me to jumpstart my career, especially in the first couple of years. And so from there, the next to that is I got a P30 development grant on AMPK regulation of tau pathophysiology, which is something I will be talking about a little today. So that's really a very important pillar of the beginning of my training, of my faculty career. And then I was awarded the Rex Scholar afterwards. However, I declined it because I got my R1s awarded in 2022. So I got a regular R1 and then an early stage investigator R1 called CATS Award. And then it looks like I'm going to be promoted to associate professor this year. And I started to take the leadership role in the ADRC T32 training program started by Dr. Koo and Dr. Glasgow. So we just started this new training program. So since we talk about T32 and since I have the mic, I want to talk a little bit about the program. So we are currently in the last year of the current funding cycle of T32. So every year we award 2 pre-doctoral trainees and 2 postdoctoral trainees or fellowships. So in a given year, we have 8 trainees that are active on the grant, except for this year because we're on the last year. So this year we only have 1 year of the funding. And so beyond just the personnel support, we try to provide some educational opportunities. And that include, as some of you saw already, there's special seminars that are invited, speakers invited from by our trainees from all over the place, and then they usually will have a Zoom seminar with us, and it's a really good opportunity for our trainees to interact with those more like higher profile scientists. And then that alternate with our neurodegeneration seminar series. We have one this morning that's usually a hybrid format right now. And so these two seminar series really, I think, are great learning opportunities for us. And then we have this Lunch and Learn series, which I was told is sort of a merge of 3 training program, and the RAC, the RIGMAC, and the T32 training program. So with the goal to encourage interaction and communication among our trainees. And then I think in the past, we also had this annual scientific retreat about among our T32 scholars, sort of as a platform for everyone to present their work in progress. It's a half-day retreat that's on campus. It didn't happen last year because we're in the transition, but hopefully once we get refunded, we'll restart these activities. All right, so that's what I had for the career part. I'm going to switch gear and talk a little bit about the research I do in my lab. So this is sort of an overview that I use for other settings. So basically what we do is we try to understand the pathophysiology of tau in Alzheimer's disease. And especially I'm interested in those crosstalks between the brain or the nervous system and other systems. So what I mean by that is that, as we know, the brain disease is not just the brain disease in isolation. There's a lot of interactions between, say, the immune system, the reproductive system, or the sex hormones, as I put here, metabolism, which is the focus of my talk today. So why do we care about that? Well, because all of these different systems also undergo dramatic changes during the aging process besides the brain. And so there's a lot of correlation, but how do those contribute to the disease pathophysiology? And is there anything we can do about interfering with those processes is something that I think are not very well parsed out. So what I use in terms of research approaches, we use this, I call it holistic approach or interdisciplinary approach, because by nature, we're looking at different systems. So we have to borrow and use other people's tools and models to study our question. The model system I talked about, we have mostly animal models, mice, flies, because I was trained as a fly geneticist, an iPSC neurons and glia model, as well as ex vivo brain size culture models. And then we had a lot of collaboration with people on campus and outside of a campus. We use cutting edge imaging technology as well as multi-omics approaches to study our systems. Now, in terms of the therapeutic translation, we'd like to test a lot of existing drugs for repurposing. And so we have some successful examples in the past, so we're still pursuing that angle. And then dietary intervention, as well as hormonal supplements. So those map to our current research focus in terms of sex hormone and metabolic regulation. Now, of course, we're also interested in identifying new targets for intervention. So I want to mention that I'm not going to be talking about the sex hormone aspect of the research, but I want to mention that that really it started by when I first started at UCSD. I had a chat with my colleagues, Sarah Banks and Aaron Sunderman here. We had coffee and we're talking about, their work and my work. And that's where everything started. We're talking about, okay, what if we look at the sex hormone, especially the male hormone in their role in the female tau pathogenesis. So they are doing the human side of the study, right? And they were like, can you test that in your models? So that really become a seed of a couple of the pilot grant I was awarded, a CTRI grant, which leads to that CAS award. So I think that's the story that I'll talk about next time, hopefully. And we have some pretty surprising but exciting findings. But for today, I'm going to focus on the metabolic story. So why do we care about metabolic regulation? Well, because we know AD is an aging-related disease, but during aging, there's also major changes in metabolic dysfunction. And in fact, it's reported that 80% of people with AD have some sort of metabolic dysfunction, for example, insulin resistance. And we also know that there are many shared features between metabolic disease or metabolic dysfunction and neurodegeneration, and that includes lipid metabolism impairment, mitochondrial dysfunction, oxidative stress, and inflammation. And so our overarching goal is to understand this interconnection between metabolism and neurodegeneration to see, what's the underlying mechanism and to explore intervention strategies. So I already talked about the approaches we take. We use imaging and multi-omics in combination with our pyramid of model from mouse to flies to cells. And I should mention, I also use the ADRC human brain tissue bank samples. So the first story I'm going to talk about was published last year, which has to do with lipid metabolism. So lipid metabolism, turned out, is very intimately associated with Alzheimer's. First of all, we know a lot of the genetic risk factors are directly actually involved in lipid metabolism, including ApoE and TREM2. And on the other hand, there's also so-called peripheral lipid modifiers, which are really metabolic diseases such as obesity, diabetes, or high cholesterol that are thought to contribute to Alzheimer's disease risk. Now, in fact, over 100 years ago, in Alzheimer's original report, besides the very well-known tau tangles and amyloid plaques, he also mentioned something like many glial cells show adipose saccules. So these black dots are fat staining or lipid staining. And of course, we didn't know what that is at that time, but nowadays we know these are in fact lipid droplets. So lipid droplets are actually a specialized organelle in the cells. So they're not just fat aggregates. In fact, they're very well regulated. So they have this monomembrane outside filled, and then in the inside, it's filled with neutral lipids. So what do they do in the brain? And in fact, this is not just in AD, and this is a study done a couple of years ago from Tony Weiss-Corey's group, where they reported this so-called lipid droplet accumulating microglia, or LDAM, in aging human brains. So as you can see here, those green dots are lipid droplets in microglia labeled by TMEM119. And this other study came out a little bit later. We're focusing on AD brain, and basically they confirmed what was reported originally, that there are lipid droplet-laden microglia that are coming in close proximity with amyloid plaques. So when we started this project, we were interested in how and why do we have lipid droplet accumulation in Alzheimer's disease brain, and in particular, since I'm a tau person, in the setting of tauopathies. So in order to see those lipid droplets, we had a very nice collaboration with Dr. Lingyan Shi's lab at the bioengineering department here at UCSD. And this work was headed by a very talented postdoc, Yajuan Li. So what their lab is specialized in doing is this so-called stimulated Raman imaging, or SRS imaging. So this imaging, without going into too much details, basically allow us to visualize the in situ lipid or any metabolized species based on the Raman spectrum. So no label is needed. There's no artifact induced by any lipid labeling reagent. So it's a very powerful technique. And so what we found first is that this is our tau transgenic mouse model, PS19. It overexpressed this mutant tau in the brain of the mice. And we found that in the diseased or the mutant or the transgenic mouse, when they are aged, where they have the full-blown neurodegeneration phenotype, we observe a lot of lipid droplets in their hippocampus. And such accumulation of lipid droplets are correlated with neural degeneration, in this case measured by the thinning of the neuronal cell layer. And we next correlated that, or co-registered the lipid droplet Raman signal with a fluorescent indicator of the ROS, or the redox ratio, measured by the FAD to NADH ratio. So this is two-photon imaging, which indicates the oxidative stress in the tissue. And we found that those areas that has lipid droplet accumulation are correlated with higher level of the ROS. And we also examined how those lipid droplet pathology are spatially related to the tau pathology. So here we found that those lipid droplet are hovering around those neurons that has phosphorylated tau inclusions. And we also look at the different cell types specific markers to try to figure out which cell type accumulate most of the lipid droplets. And here we found that most of the lipid droplets in those tauopathy mouse brain happen to be in microglia. So up to 70% are in microglia. There's also a good amount in astrocytes, but barely any was found in neurons. So this is again confirming or consistent with a previous report of microglia accumulation of lipid droplets. So because tau pathology, at least in our model, started completely in neurons, there's no amyloid plaque outside of the cells, we wonder, could those lipid droplet phenotype or the pathology we're seeing start from neurons? Or in other words, are neurons the ground zero of all these lipid pathology? So we decided to take advantage of this iPSC-derived tauopathy neuronal model. So this is a model developed in Lee Gan's lab when I was a postdoc there. So we have this isogenic lines of wild-type neurons and a FTD mutation carrying tau neurons. So the tauopathy neurons have more pathological tau accumulated. So we did Raman imaging on them. And what we found is that the mutant tauopathy neurons actually can accumulate lipid droplet when you culture them in a dish. So they are capable, or they have this lipid phenotype on their own. And again, that is associated with higher oxidative stress and higher level of lipid droplet associated gene expression. So now if tauopathy neurons actually can accumulate lipid droplet, how come we only see microglia or glia cell having lipid droplet in vivo? So we asked this question, how do neurons that are fat burdened talk to microglia potentially? So we did a very simple experiment, which is we collect the neuronal condition media from either wild type or the tauopathy neuron, and then we put them onto a microglia cell line, in this case, BV2 cells. And we did Raman imaging on them. And what we found is that those microglia cells, when they are treated with the mutant tauopathy neural condition media, they have a lot of lipid droplet accumulated. So next we ask, is that a direct lipid transfer from neurons to microglia, or is it indirect? So to do that, we took advantage of this technique called heavy water labeling technique in conjugation with the Raman imaging. So the idea is you can label any newly synthesized metabolite using a deuterium water, so D2O. It can label any metabolite with the heavy water or the CD bound, which can be picked up by Raman imaging, which allow us to trace those newly synthesized metabolite. So what we did is we label our tauopathy neurons by culturing them in heavy water containing media. So that will allow some labeling of the heavy or the deuterium bond in deuterium to carbon bound in the lipid. And then we change the medium out to a normal water containing media, allowing those newly synthesized lipid be released into the normal water containing media. And then we use this media to treat our microglia. So what we found is that not only do those microglia accumulate lipid droplet when they seize the NCM coming from the mutant tau, but now they actually pick up the heavy water or the deuterium signal. What does that mean? These microglia has never been cultured in the heavy water. So the only source of the heavy water labeled lipid is coming from those neurons that they were pre-labeled with the heavy water containing media. So this is a direct proof that at least some of the lipid that we saw in microglia comes directly from those neurons who are dumping their lipid into the media. Now, what does it mean to have lipid droplet accumulating microglia? So we took a look at their inflammation profile by measuring the pro-inflammatory versus anti-inflammatory cytokine gene expression. And we found that those microglia treated with the mutant neuronal media that induced lipid droplet accumulation have increased pro-inflammatory cytokine. And moreover, when we did this phagocytosis assay by applying this fluorescent labeled beads, we found that those lipid droplet accumulating microglia have reduced activity to phagocytose. So these matches the so-called LDAM phenotype, as they call it earlier. So they're supposed to be disease associated or disease contributing. Now, this is what I call the moral of the story is sharing is caring unless it is fat. In this case, we are talking about actually two stories. On the one hand, this is the two work that I mentioned earlier, that they started from A beta model, in which case they saw that A beta extracellularly can directly induce microglia lipid droplet accumulation in an ApoE-dependent manner. And so interestingly, they did this reciprocal experiment as I did. So they put the microglia conditioned media from these lipid droplet bearing microglia, put it on neurons. And what they found is that that can induce tau pathology and neurotoxicity. So what we found is the other side of the story. So if you have tau pathology to begin with, we found that neurons actually have this lipid burden, and they would dump it out, give it to microglia to handle, and as a result, those microglia will accumulate more lipid droplet and come back to cause more tau pathology and more neurotoxicity. So this is creating this vicious cycle. All right, so the next question we ask is if neurons can be the initiator of this whole fat pathology, how does neuronal metabolic signaling regulate such a phenomenon? So here comes to the AMPK story that I mentioned earlier. So first of all, let's think back of the brain metabolism as a whole. So this is a figure that I cite all the time. Basically what it shows is that in an AD brain where you have higher tau accumulation by the PET imaging, you also see this region have the low glucose uptake, okay? And so that's a very nicely correlated phenomenon. So one of the hypotheses that we had is that maybe when the brains are suffering from lower glucose metabolism, they have to switch to a different fuel source, right? In this case, lipid. So for those neurons, they can metabolize lipid. However, that will create a lot of free fatty acid lipid burden. And neurons do not like that. They don't know how to handle that. And what they do is that they will accumulate lipid droplets as a way to protect themselves, and or they will dump it out to the glia, astrocytes, and microglia, and they will handle it. And that's why we're seeing those lipid droplet pathology in the brain. So this hypothesis led us to take a look at this enzyme called AMP kinase. Okay, so AMPK, as we might all know, is a master regulator of energy metabolism. So it senses the cellular or tissue energy level. So it's activated when you're starving, we have a low ATP. And what it does, it's regulating both the glucose metabolism and the lipid metabolism. So when we took a look at our ADRC samples that has BRAC stage 1 versus BRAC stage 6, we found there seems to be a decrease of AMPK activity in those advanced disease brains. When we look at our iPSC neurons that I showed earlier that has the lipid droplet phenotype, we also found a very significant reduction of AMPK activity. So with that, we decided to treat our neurons with the AMPK activator to see how does that do to their lipid droplet phenotype, which we established earlier. So we treat our neurons with this AMPK agonist, ACA. So here shows that in the mutant tauopathy neurons, where there's a lot of lipid droplet, the ACA treatment leads to a significant reduction of lipid droplets. So that suggests there's a cell autonomous effect of neuronal AMPK in regulating or in inhibition of their lipid droplet accumulation. And we also show that it's through both reducing lipogenesis in the neurons and enhancing lipophagy, which is a form of autophagy that promotes the lipid droplet turnover. And what about microglia? So we took neuronal condition media from those neurons that are pre-treated with this AMPK activator. And now we use the condition media to treat the microglia, the same assay that we did earlier. Now, those neuronal condition media can no longer induce lipid droplet that we saw earlier. So this suggests there's also cell non-autonomous effect of neuronal AMPK in regulating microglia lipid droplets. And of course, we want to bring this back to our in vivo mouse model. So in this case, we are going back to our tau transgenic mice, and we genetically deplete AMPK in one side of their hippocampus using a virus. And then on the other side, we injected tau fibrils. And we tried to look at tau pathology and lipid pathology, lipid droplet pathology on the AMPK manipulated side of the brain. And what we found, as shown here, is that when you deplete AMPK, you have a higher level of tau aggregation or tau spreading. And you have higher level of microgliosis, shown by CD68, as well as higher level of lipid droplets accumulation. And the lipid droplet number correlates with the tau pathology. All right, so that was the first story that I meant to share. So this was published last year in Cell Metabolism, and here are some highlights. And I do want to give special thanks to ADRC for the human brain tissue samples for the development project, which I got the first, it's my very first grant, to be clear, that really jumpstarted this AMPK project, and all the awesome collaborators that I know through the ADRC. All right, some open questions. Of course, we're also interested in AMPK in microglia. In fact, I proposed that in my development project. I want to do that on both sides of neurons and microglia. So we're exploring that and other signaling pathways that regulate lipid droplet metabolism. And of course, we're also interested in other cell types beyond neuron and microglia, such as astrocytes and oligodendrocytes. Now, besides this side of story, we also wanted to explore, can we target lipid metabolism as a strategy for intervention in tauopathies? So this is what I call the other side of our fat business, which I'll just talk about very briefly because this is still a project in development, that we are focusing on ketogenic diet. So why ketogenic diet? Well, as we know, this idea of eating for brain health has been really focused these years. And we know that the difference between the so-called standard American diet, which is heavy on carb and fat together, there's these so-called metabolically healthy diet that have different compositions of their macros, but there's one thing in common, if I could point out, is that they all induce ketosis, at least at some point of the day. So it could be a high-fat diet, it could be a high-fat, high-protein diet, it could be a caloric restriction, or it could be intermittent fasting. So what happens is that during the day or during some course of the diet intervention, there is ketosis, there's keto body being made. Now, in fact, this keto as a neuroprotective agent has been explored for Alzheimer's disease. There are ongoing clinical trials. Some of these might have been finished, and they are giving some promising results in terms of improving cognitive function, improving AD biomarkers. However, they're also faced with challenges such as compliance, and there's major side effects associated with this high-fat diet. So what we're trying to do here is to really improve the ketogenic diet if it's promising. And to do so, we'll need to understand how it works to begin with. So in order for a ketogenic diet to work, the active component is beta hydroxybutyrate. That's the major keto body that people talk about, has to be made, okay? And in order to induce BHB production, one could either follow those dietary regimen I just talked about, or one could provide exogenous BHB supplementation, such as giving them precursors or injection or feeding. And so by doing so, we try to A, phenocopy the effect of ketogenic diet, and B, understand the molecular mechanism, or what is the molecular mediator of BHB in terms of its beneficial effect. So the strategy we took is to use this keto precursor called C6X2BD diet. So basically this compound, when it's metabolized in the body, will turn into BHB. And instead of relying on very high fat or very low carb diet, we can now basically supplement a normal diet or controlled diet with this compound to achieve ketosis. So this project is led by Liana Rosito, a very talented graduate student in the lab who is a current T32 pre-doctoral fellow. So what Liana has been doing is to treat our tau transgenic mouse model PS19. And she fed the mice for 3 months and collect their brain at the end of the 3 months and fraction their brain lysate based on their solubility. And what she found is that I'm only showing the insoluble fraction of the brain lysate. But basically you can see here is that across all the different fractions, there is the significant reduction of pathological tau species, in this case, a phospho-tau labeled by 88. Besides, if you look at the higher molecular weight, which represent those oligomeric aggregated tau, you also see a striking reduction with the diet supplement. When we look at their pathology in the brain, I don't know if you can see on the screen, but basically here is we're labeling the tau tangles by this MC1 antibody. And again, we're seeing a very striking reduction of tau tangle. And that is accompanied by a reduction or amelioration of neural inflammation as labeled by GFAP and IBAL1 staining. And we also did this fibril-induced tau spreading model in which we inject the mice with a synthetic fibril on the one side of the brain and put them on a diet, and then look at how much tau is getting spreaded to the other side of the brain and how much aggregates we're seeing. And here, again, we are seeing a significant reduction of tau spread or tau aggregates in response to this fibril inoculation when they are put on a keto diet. Okay, so the question we wanted to ask is like, how does BHB actually achieve that impact on tau, pathological tau proteostasis or tau turnover? So if you think about the proteostasis of tau, there are several aspects cellularly. One, tau can seed. A bad tau can seed good tau, and that leads to this tau pathology formation. And tau can be secreted from neurons, and then tau could be uptake from outside into the neuron. And that creates this cycle that allow it to propagate. So I'm not showing the data here, but we have some pilot data showing that BHB can inhibit all three aspects of the cellular behavior of tau, which are involved in their propagation. So we really want to understand how is BHB working to affect the intracellular behavior of tau. So the approach that we're using is to take this systematic biology view to to look at the protein-protein interaction network of tau between the control condition and the BHB treated condition. So the idea is if you can see where did tau go in terms of their interaction, protein interaction network in the neurons when they seize BHB, that can inform us of how BHB actually regulate tau proteostasis. So to do so, we took this proximity labeling approach, which is APAX tau. So APAX is the enzyme that have been engineered onto tau, and it will label any protein that's in close proximity, 200 nanometers, to in the cell lively, and then we can hold down those labeled proteins and run quantitative mass spec. So this project was in collaboration with Sam Myers and his graduate student, Mel Brunelli, at the Hoya Institute of Immunology. So I'm not going to show you all the data, but this is a very broad overview of all the hit we found. I know it's a lot of information, but I want you to see these big dots here. So we pulled out a bunch of interactors in the cytoskeleton, synaptic vesicles, cytosolic vesicles, and cell junctions. We also pulled down things in the nucleus, in the ER, and in the mitochondria. So what's interesting, there's a pattern here. What we found is that BHB seems to enhance those tau interactions with its normal locations, or its normal housekeeping functions, and with proteostasis pathway components. So that includes cell skeleton, because tau is a microtubule binding protein. So we see increased tau interaction with cytoskeleton, and we see increased tau interaction with those vesicles, and that includes autophagy vesicles and a lot of the vesicular transport proteins. So that suggests this could be the reason why we're seeing increased tau proteostasis or enhanced tau proteostasis. On the other hand, BHB seems to reduce tau interaction with those, what we call ectopic location of tau. So tau has been repeatedly reported to present in a nucleus and in ribosomes in mitochondria, and those are considered abnormal location, okay? So they could be there, but if you have too much presentation of tau there, it's probably doing some harm. And we do not know the specific pathway and what's behind that, but we do see is that BHB seems to reduce those interactions. So we're currently trying to validate some of the targets and try to confirm and dive deeper into the molecular pathways that's underlying such changes. But in summary, for this project, we found that BHB supplementation can ameliorate tau pathology in mice, and BHB can promote tau proteostasis, inhibit tau propagation. And our tau interactome study showed that BHB alters the tau protein-protein interaction network in neurons. So in the future, we're going to look into more of the molecular mechanism. For example, how much of this effect of BHB is attributed to its role as an energy source or as a fuel versus as a signaling molecule. We want to dive deeper into different cell types effects. So we have a parallel project on microglia response to BHB, and we just started to look into astrocyte response to BHB in the setting of tauopathies. And ultimately, we want to understand the molecular mediator and pathways and hopefully identify more potent treatment strategies. And that brings me to the end of my presentation. I'd like to acknowledge my lab. So these are my current lab members. I mentioned Yajuan Li. She is actually a postdoc in Lin Yanshi's lab. She was a visiting postdoc in my lab last year, 2 years ago. And she was the first author of the lipid droplet study. And the ketogenic diet study, as I mentioned, is led by Liana. And I have collaborators. Some of them are from ADRC or affiliated with ADRC, and the funding source. And thank you very much for your attention.